Okay, guys, it's going to start on chapter 16. We're going to talk about epidemiology. Epidemiology it's kind of like the detective branch of medicine. It's where we track down the causes of disease and also includes a lot of statistical analysis, frequency. The two main things, and these are definitions, so yeah, I would know this if I were you, is incidence and prevalence. So if I'm talking about the year 2019, well, no, let's talk about this year, 2020. We won't do coronavirus because that's kind of in the thing. Incidence is a new diseases, new cases. This is the key thing during a period of time. So in 2020, the new cases of HIV, that's what incidence cares about. What about all the people who still have HIV who got it in 2019? When incidence is concerned, I don't care about them. They don't count. Prevalence is the total number of diseases. So if there are people who got HIV in 2001 and they're still alive, they count for prevalence, and everyone who is alive that has the disease is prevalence. Total. Incidence is new cases. Okay? That's the way we use those two measurements. Occurrence, we also evaluate it by how often it's occurring and where it's at, its distribution. We can also use these to describe the morbidity. In other words, the effect the disease has on reducing function among individuals, and mortality, the number of deaths resulting from that. Here's our curves, incidence and prevalence of AIDS, which is full-blown HIV. So they're not telling us, oh yeah, they are. Okay, so the gold line here is the incidence in new cases. The new cases peaked somewhere around 1993, 94, and then they've been on a downhill slide and went back up around 2004, and they started going down, went back up, and coming back down. But they have taken a steep decline. So the new incidences of disease are decreasing. Prevalence has gone way up in the same period of time. What happened with that? How can I have more prevalence if I have less incidence? Well, the short answer is people are living longer. In the beginning, HIV, the incidence and the prevalence were very close together because new cases of disease only lived a very short time before they died. So the longer we're able to keep people alive with the drugs that we have, the greater the prevalence is going to be. Of course, with the incidence going down, at some point, we hopefully will see prevalence going down as well. Okay, patterns as far as where they're at. Endemic disease, these you see sporadic, no, not sporadic, we're going to use that term. We see widely separated cases of disease all the time. It's never totally gone out of the population. TB. Tuberculosis is endemic. Other parts of the world even worse, but there's always going to be cases of tuberculosis in the U.S. and North America and South America. Okay, so that's the endemic diseases. Low level, but always present diseases. Sporadic, not always present, just kind of randomly pops up here and there, but not many cases. Epidemic, you get a lot of cases in a certain area. Pandemic, which everyone is familiar with now, you get a lot of cases in a lot of different areas. And some pandemics are, of course, like coronavirus worldwide. Disease causation, we already talked about etiology. This is one of the main things the epidemiologist wants to find out is what's causing this, because if we can find out what's causing it, we may be able to stop the disease by limiting people's exposure to its causative agent. Connecting the disease to a certain pathogen, that sometimes is going to be really a challenge. Because we need to do direct causation as rather simple association. There is a bacteria, no, excuse me, let me think. There's a virus that is called pneumoniae because it was always found in people who had pneumonia. But it actually did not cause the pneumonia. It was there. It's always associated with it, but it wasn't the cause. The cause was the bacteria pneumococcus. So that's kind of what part of the problems. Just because something is in an area at a certain time doesn't mean it's the one that's causing the problem. So if I were standing outside of a bank and the bank got robbed and the police came and arrested me because I was in the same area as the bank, see, that wouldn't make any sense. We have to be able to prove the direct that this is the one that's causing the disease. Okay, as far as our public health organizations, nationally we have the United States Public Health Service, of which the CDC is a branch. We also have public health at the local and the state level. We have the United Nations sponsors the World Health Organizations, and these guys are supposed to be coordinating public health worldwide, while the CDC does it on the national level. Well, I'm not going to kick in as to whether either one of these guys are doing a real good job. People are people. The CDC does publish this, and I always found this interesting. It monitors and lets you know about prevention and other reportable, notifiable diseases. It publishes it online in the Morbidity and Mortality Weekly Report. They also, in that, they will give you information regarding, say, for example, people get staph infections, folliculitis, boils, I guess people would call it. And, you know, they will give you information on which antibiotic has been shown to be effective against this in this specific area. And that specific area could be part of a state or part of a city or part of a region like the Southeast. And they give a lot of valuable information. They're working to try to limit disease transmission, and this is one of the biggest things. The reason that people are living so much longer, the lifespan has expanded, is not due to we physicians treating people after they get the disease. It's due to these guys, the epidemiologists, which are physicians, by the way, and the health inspectors too. The guys who actually force people to put their septic tank X number of feet away from their house, and the people that go in and inspect those restaurants that we are prone to eat in. They work to try to find out what's transmitting the diseases. Reservoirs are places where the diseases hang out. For example, there's a deer tick that is a reservoir for Lyme disease. that's where they hang out at. Well, the tick is going to be the vector. Reservoirs would be something like the prairie dogs. Prairie dogs are reservoirs for the plague. Armadillos are reservoirs for leprosy. These guys establish and, well, the enforced part is a little bit up in the air, but they're the ones who give us the guidance on when and at what time to, well, when and how to administer vaccines. Locate and treat individuals exposed to contagious pathogens. Well, really more locate than treat. Establish isolation and quarantine measures. I feel like I'm kind of preaching to the choir because you guys have been living through this for the last few months, but we'll go through it anyway. Descriptive epidemiology. These are the guys that are going to go back in detective work and going to get all the location and time of disease cases, going to collect all the patient information, identify the index case, which is usually going to be the first case. In doing this, they're going to develop a theory, hypothesis of what's causing it. It could be tested by experiments such as the animals, lab mice. They're showing this outbreak of cholera. They were able to track the cases down, and in doing this descriptive epidemiology, they were able to find out that there was one certain water pump that all these cases of people were using. See, there are all these pumps here. And I believe it was this one right here was the pump that all these people that died from cholera had gotten their water from there. Analytical epidemiology, probable cause, mode of transmission, methods of prevention. These are the guys that are doing the research to find out what's most likely causing it, how it gets passed around, and what we can do to stop it. And this is good where you can't really use the Koch postulates. Now, guys, this is hindsight. We're looking back. This is after everything has occurred. We can look and find out so we'll be able to prevent it the next time. That's just the way it is. Experimental, this is where and we talked about this, they get once someone develops a hypothesis in descriptive or analytical, these guys are going to use the lab animals and test it. These guys will definitely use Koch postulates if possible. How do we get diseases passed from, we talked about the portals of entry and exit, but how do you actually get these guys moved around? Well, from a reservoir or a portal of exit from one person to the next person's portal of entry. 3 ways: contact transmission, vehicle transmission, or vector transmission. Okay, direct contact transmission, that's when people are in contact with each other. And actually, this can actually occur in a single individual. If you have an infection, say, on your finger and you're constantly rubbing your eyes, you can actually inoculate the bacteria and cause an eye infection. And this is something you see a lot in toddlers. They don't know any better than to touch something dirty on themselves and then put it someplace else. Some adults, of course, too. Indirect, this is by fomites. And let's use the toddlers again. In the morning, Johnny is at the daycare. He grabs a toy, puts it in his mouth, and slobbers all over it. He sets the toy down and leaves at lunch. That afternoon, James comes in. Let's call him Jimmy. Jimmy comes in, picks up that same toy, and puts it in his mouth and slobbers on it. that toy is a fomite. Anything that is non-living, that is a way for microbes to be transmitted. And that's really famous as far as, and we know this today with our coronavirus, that we need to disinfect things like doorknobs and things, or wear gloves or both. And that's what a fomite is. It's an indirect, because one person touches it and someone else comes along and touches the same object. Droplet transmission, droplets of mucus by breathing out or coughing or sneezing get big droplets that contain the whatever pathogen virus. And look at that area, and that's not a really big sneeze. A sneeze or a cough will spread out lots. Now that person probably actually had a mouthful of water before they did this, but the point is to give you an idea of the range, not the quantity. Vehicle transmission, airborne, waterborne. So these aren't fomites, but just actually things like the air or water. Airborne, when the pathogens travel more than about 3 feet through aerosols, this can be sneezing, coughing, an air conditioning system, or even sweeping a floor. Waterborne transmission, a lot of GI tract infections occur this way. Guys, diarrhea is almost always fecal oral. So some sort of fecal matter got into your mouth and into your system, and that's what caused the infection. Okay, vehicle transmission, foodborne, pathogens in and on foods. A lot of times the food isn't processed or cooked well enough or refrigerated. And we know about this. They're always recalling food for E. coli contamination. Foods can be contaminated with feces. Again, it's kind of gross, but it's true. Body fluid transmission, blood, urine, saliva, and semen can carry many different kinds of disease-causing organisms. What we have to do is to keep from coming into contact with a person's conjunctiva, remember the tears, breaks in the skin or in the mucous membranes. Those are going to be routes through which body fluid transmission can occur. Food transmission, poorly refrigerated. Unfortunately, guys, I mean, the rest of the world does not live with the standard of luxury we do, and these poor folks, this is all they can do, but it does increase their risk of foodborne diseases. And honestly, this is maybe something we won't see again in our lifetime. This is probably going to be a passé thing. All the food bars that used to be where people would come up, we'll have to wait and see after the coronavirus is over. I'm not sure what's going to happen with that. And finally, vector transmission. This is where an animal or an insect, which is an animal, can transmit the diseases among hosts. Got the biological vectors. Here's the fly. Flies around this dog droppings and even lands on your hamburger. You eat the hamburger, so you get sick. Kind of gross, but it happens. This is one that happens very frequently, especially in the tropics with all the malaria and everything. The mosquito that is infected with the malaria protozoa bites a person, injects, and while they're sucking the blood out, the malaria larvae kind of get into the person and then go into the red blood cells, and they reproduce, they burst the cells open, and they just continue the cycle. And then another mosquito comes and bites that person that has malaria, and now that mosquito has malaria to bite another person and help it spread. That's why one of the best ways to prevent malaria is to kill mosquitoes or protect against them. So the biological vectors, um, these guys are going to serve as a host, either temporary or permanent, for the bad guys at some stage, such in the malaria that goes through several stages. Biting arthropods transmit many diseases. Arthropods, think ticks, Rocky Mountain spotted fever, Lyme disease. Mechanical vectors passively transmit pathogens present on their bodies to new hosts. Not really microbiology, but think lice or scabies. Okay, guys, quarantining. Again, we know a lot about this nowadays. We've all been in quarantine. We're starting to come out of it, hopefully not too soon. Although I'm pretty sure I can speak for everyone who said we're all tired of it. But it is what it is. Individuals suspected or known to have been exposed to a contagious, remember contagious is easily transmissible, may be quarantined or basically isolated until they are completely recovered. Remember, we are actually contagious during all stages of disease, even recuperation. Usually, we do special areas for this. Tuberculosis. Even before the coronavirus, when we get a patient that has active tuberculosis, we put them in a room. We require masks and gowns on all the persons going in there. Not only that, but we also use a negative pressure room. What does negative pressure mean? It means that we have the air pressure in there lower than everything around it. So which way is the air going to move? It's all going to move into the room and none of it is going to move out. As far as venting the air, we can actually take that air in the room. We run it through a filter, one of those HEPA filters, and that way we prevent the spread of tuberculosis. But yes, we do have to go through extra precautions. Ebola, oh my goodness, that was a nightmare too. There you can see in Nigeria an Ebola ward. Here is one of those things that they use to transport highly contagious patients. And I guess some of those nurses that they brought back from those regions of Africa where Ebola were. Special air handling methods, special protocols, what we've already talked about. Nosocomial, also called healthcare associated infection. Both of those are just two words for the same concept. This is the truth. There are some nasty microbes in a hospital. One of the nastiest places on earth is the hospital. Talk about another truth. People in the hospital are sick. They're immunocompromised. They're susceptible for infection. Now, the only thing that we need to make this happen is for us to pass the bad guys between nurses and doctors and patients, or patient to patient. A lot of hospitals are going to private rooms, and I think in some ways that's a good idea, even though it's more expensive, because it keeps you from getting sick from the person in the bed next to you. Now what we have to do is make sure that our staff, the nurses and the doctors, do not carry C. difficile from patient in room 201 to Granny in room 202, because Granny might not survive it. So that's what we have. We really can't do very much about this, guys. They do their best, they really do, but it's just the nature of a hospital to have bad bugs that are there. Can't do anything about that. People are in a hospital because they're sick. That's why they're there. This is the one that we have total control over. We have to make ourselves. Here's what I always do. I would do my hands as I come into the room, use the alcohol foam mostly, deal with the patient, and of course, if the patient was actually contagious, I'd be wearing gloves. And then on the way out from the room, use the alcohol. And if someone had C. difficile, you have to actually wash your hands. But anyway, types of healthcare associated infections: exogenous, that's where you get it from the hospital. Endogenous, it's where normal flora actually becomes pathogenic. And I already told you about that. That's when you leave the Foley catheter in too long, you leave the IV in too long. Patients do not like for you to pull out the IVs and put it in a different location. That's understandable, but that has to be done, because if we leave it in place too long, several days, the bacteria will make their way into the patient's blood, because that's where the IV is going. Or the bacteria will crawl up the plastic Foley catheter and wind up in the bladder and cause a urinary tract infection. We already talked about iatrogenic superinfections when we use the antibiotics and kill off normal flora. We talked about that when we talked about C. difficile. Emerging and re-emerging diseases. We monitor and prepare for emerging infectious diseases such as the Ebola and the Marburg. These guys have not historically been an issue for healthcare, but they are starting to come out and starting to spread. At one time, HIV was an emerging disease. Now, how long was it actually there before we noticed it? Who knows? HIV could have been floating around in the 1940s. I don't know. No one does. We noticed it in the 1980s, and that's when it was an emerging disease. Emerging is either going to be totally new, or it's going to be an older disease that all of a sudden makes a comeback. That's what happened with tuberculosis. There for a while, we used to have whole hospitals, especially in our state, devoted to tuberculosis. We had in Rome, Batty State Hospital was for TB only, where patients were taken and then were sort of isolated from other folks. And TB, with the emergence of new and powerful antibiotics, was almost wiped out. There's big underline on that, almost. It wasn't, and people got very lax in taking their antibiotics, and so it came back drug resistant. So that's the re-emerging. All right, guys, that's got it for this one. It wasn't as long as that chapter 15. And we got one more, and that'll be everything for week 5 of the semester. Thanks.